This is a production of Cornell University.
It's a pleasure to be here at Cornell for many reasons. There's a movie called Ghostbusters in which they are trying to make a character as nerdy as possible. And this character says, I love mold spores and fungus, and so do I. So I like to start my talks with that so you know where I'm coming from. Here's the plan. I'm going to spend a lot of time in introduction and then go on and talk to you about some of my fairly recent research and then wrap up with some generalizations. So it'll be a shaggy dog introduction. I spent most of my career at Tulane University, which has the advantage of being in New Orleans, Louisiana. It has what's called a geographic endowment. A lot of people go to Tulane because they want to live in New Orleans. It's a wonderful city. It's often described as the most European of American cities, but that's not true. It's more an Afro-Caribbean city. A lot of voodoo and it's a very Black city. It's a wonderful city, special environment, and it's the birthplace of jazz. Fabulous food. Anyone who's been there knows how hard it is to keep from gaining weight. Actually, the coffee house culture goes way back before Starbucks and is still going strong. And if you've ever been to New Orleans, you know about the wonderful beignets, carnival, and all these things come together. Here I'm a girl who grew up in Brooklyn, New York, and I fell in love with New Orleans. And I think of all the things I love about New Orleans most is there's something about the people and the attitude and the way people interact. The city, you can't live there without picking up some of it. Now, having said this, while I was in New Orleans, I did research on one of these nerdy fungal topics. I studied mycotoxins, especially aflatoxin, which is usually introduced as a singular, but it's a family of compounds. I've given you the structures of the major ones, and they're made by several genera of Aspergillus, a major fungal genus. In as much as I have a scientific claim to fame, it's that I isolated a lot of block mutants in the aflatoxin pathway, and I was lucky. Many of these block mutants turned out to be colored, and we, with great difficulty over the course of more than 20 years, laboriously worked out different steps in the pathway. And when gene cloning came along, we were able to discover that the steps were linked. And this became a paradigm for other mycotoxins and for other secondary metabolites, that these unusual small molecules tend to be biosynthesized by these clustered gene pathways. And those of us who work on aflatoxin are really proud that we beat the penicillin people. Just think of all that industrial money for all those years, and we happened to get there first. Certainly, I did not do the work alone. I isolated a lot of the mutants. Much of the gene cloning was done by Peng-Quan Chang and Zhu-Jiang Yu. Nancy Keller, who's a graduate of Rutgers, was deeply involved. Ed Cleveland was the director of the group, and there were other people, especially Deepak Bhatnagar and Gary Payne's group. I'm the one, I'm lucky, I got a lot of the credit, but it was definitely a group effort, as science always is. Now, if you know anything about fungal toxins, and I expect many of you here do, they're a big problem in the global food chain. Without a doubt, fungal toxins are bad. They infect our human diets, they infect our animal diets, and they cause a variety of terrible diseases when ingested, and depending on the toxin, different problems. More controversially, mycotoxins have been blamed for certain building-related habitats. When people live in moldy buildings, and this is a sort of famous picture in the world of indoor air. I mean, that's a really moldy building. But when people live in contaminated habitats, it's been hypothesized that they are involved in something that's generically called sick building syndrome, not a good name. A bunch of these non-specific symptoms. There are lots of things that can give you a sore throat or that can make you fatigued or have a headache or cause insomnia. And there's this idea that when they all happen together and you're living in a mold contaminated house, they're caused by exposure to fungi and perhaps to these toxins. My perspective has been for a long time that although mycotoxins really are a problem when they come through the diet, it's hard to be exposed to enough of them through the respiratory system and enough of any one toxin to cause this variety of problems that I don't think they are the cause. Mycotoxins are the cause of sick building syndrome. Fungi might be, there might be allergic reactions. And I actually sent to college one of my sons to an expensive private college by taking consulting jobs for insurance companies where I would say, hey, this person is sick, and yes, mycotoxins are terrible, but I don't think they're sick for that reason. And I don't like consulting, but he graduated from the University of Chicago without a scholarship and without any debt because I worked for the insurance company. Okay, so I'm saying all this to tell you I knew a lot about sick building syndrome. And I've told you I lived in New Orleans. And many of you are too young to remember this, but there are many of you who I think do. A really bad hurricane hit New Orleans in 2005. And here's a picture that was published on the internet. And I saved the newspaper. When I went back, this newspaper was that says Katrina Takes Aim was lying on the breakfast room table in my breakfast room. And I'm sorry I didn't take a picture of the paper with the chaos around it, but I did save the paper. So let me tell you a little bit about the topography of New Orleans. New Orleans is between the Mississippi River, which is in this image circling around as a ribbon going up and down, and then below a very, very large inland lake that most people haven't heard of. After the Great Lakes and the Great Salt Lake, it's the biggest lake in the country, Lake Pontchartrain. And much of New Orleans is below sea level. So this topographic map shows the areas in blue that are below sea level, that when the various levees broke from the lake, filled into the city. The Mississippi River behaved itself. It was Lake Pontchartrain that spilled. And this engineering diagram tells you what happened. The city basically filled to sea level, and everybody found out where they were in relationship to sea level. This is how it looked from the sky. You can see the Mississippi River there. 80% of the city flooded. That's 4 out of 5 people. Neighborhoods, you can see the trees here. The parts of the city nearest the river didn't flood, but most of the city did. Anyone who knows about mold knows when there's that much water, fungus is going to grow. And here's a cartoon that came out not that long after Hurricane Katrina. When the waters subside, the problem's going to be mold. And it was. There's one image that I found on the internet of post-Katrina New Orleans. Zhujiang Yu, one of the people who was involved in the aflatoxin work, was one of my few colleagues who took a picture of his home. Look at that. Look at the piano. He had flooding all the way up to the ceiling. Our home had also flooded. We'd evacuated before the storm. The National Guard wouldn't let us go home. So we didn't get back for 5 weeks. And my husband's from New Jersey. We evacuated to Jersey. I called a buddy at Rutgers University, said, can I make some media? I'm going to fly home and I'm going to sample my house. And so I did. And here's the entryway, antique clock. You can see the different places on the wall as the Army Corps of Engineers was pumping out the city. Here's the living room. There was no longer any standing water, but you can see where furniture had been knocked over. Here's our family room. I had made these pitiful attempts. I'd put a lamp up on a table thinking this room might get a little bit of water because it was the lowest in the house. I mean, it was bad. There was mold everywhere. Here's an ornamental Psidiomycete, which is just these, when I cultivated that black stuff, it was all pure Aspergillus niger. And I went into this hoping to find a new species of Aspergillus or a new mycotoxin or something. And so I sampled away. What the pictures can't tell you is the way it smelled. I was a good girl. I was wearing an N95 mask. I was wearing gloves. I had long sleeves on because I knew I was in a moldy house. And as I took the pictures, I kept feeling sick, smelled nauseating. And I'd go outside and I'd rest for a while and I'd sample some more. And I became a convert. I said, you know, there might be something to this sick building syndrome. Something is really wrong with being in this house. And I'm wearing a mask. And I know a lot about mycotoxins and I know a lot about spores. And I said, you know, they put out gases too, volatile organic compounds. And so exposure to the mold in my house is what gave me this, what I call conversion experience. Well, meanwhile, Rutgers, where I'd been making the media and brought the fungi, offered me a job. And so the day after I got tenure at Rutgers, I called my dean at Tulane and resigned my tenure at Tulane, and I moved late in life. I mean, I could have retired. And I spent a lot of my early years working on the Office for Women in Science, which was gratifying, but I also started this work on the volatile organic compounds. So that's the shaggy dog introduction. Now I'm going to give you a mini lesson on volatile organic compounds. Definition. Low molecular mass. They're little molecules. Look at those little molecules. And they easily vaporize. And any of us who are scientists know about it. Benzene, ethanol. You leave ethanol out when it's not covered, it evaporates. Toluene. Our solvents tend to be that. And then anesthetic gases. We've all heard of ether. We've all heard of chloroform. Many of the synthetic chemical compounds are known to be air pollutants, and the Environmental Protection Agency and American Cancer Society have good science that tells you about these synthetic volatiles. Less is known though about the biological volatiles, and I didn't know this when I started, but everything we smell is in the volatile phase. If you can smell it, it's a volatile. That's how our olfactory system works. And hey, you know, the biggest group of human genes, olfactory sensing genes, I'd never known that. They not only come through our respiratory system, but they can be absorbed through the skin. And this is one of my buddies at Rutgers, Charlie Weschler. He is very interested in volatile compounds, and he did this experiment in Denmark with some buddies where they went in wearing shorts and they breathed clean air, and they had a volatile brought into the room. And then they measured all their pee and poo for a couple of days. And they found that this particular compound, I'm forgetting which one it was, that 50% of what they were absorbing was coming in through the skin. So when we think of volatiles, I don't think we necessarily think that our bodies are absorbing compounds, but it's not just inhalation. We can get them from a various variety. When you read the research, it's kind of fun. I've read some book on perfume. Some of the very best research has been done by perfumers, and they keep looking for this human pheromone that'll make us sexually attractive to whoever it is we're trying to. I don't know if they succeeded, but if you've ever bought an expensive perfume, you know, they make a lot of money. We know smells. Smells are actually hard to put a name on, but often we can recognize things like vanilla or ginger or lemon. Here are some of the fungal volatiles that we all tend to recognize. Geosmin, which is also made by bacteria, is that earthy smell. Very low quantities give that odor. The one I've been working mostly on, mushroom alcohol. Limonene is also made by some fungi. And they tend to have very low odor thresholds, teeny tiny amounts. We think there's a lot there. And this makes them incredibly hard to study. You think there's a lot there and there's very little. And when you start studying them, you discover what fungal volatiles you get have a lot to do with what the fungus is growing on, because many of them are just breakdown products of the substrate. And so the term volatome or volititome, much of what's observed there is not necessarily a metabolic product of the fungus, it's a metabolic breakdown product of the fungal's enzyme that's breaking down something else. That said, there are some famous fungal odors. Stinkhorns, if you've ever gone collecting mushrooms, you may know just how really bad they smell. They smell terribly bad. And then there's perhaps the natural product that is thought to be most likely to be a real aphrodisiac, truffles, and they have a very special odor. Interestingly, people who've been studying it have discovered it has to do with the bacteria that live in the truffle, not really with the truffle, but that's another story. The people who've done the best work in signaling are chemical ecologists, where they're often called infochemicals or semiochemicals, and there's some really, really good insect work on volatiles. And from my perspective, remember, I told you I'd known a lot about sick building syndrome.
Just gonna close that up there.
What are we doing?
Just closing that up there so that they can see your slides.
Okay, sorry. People in Scandinavia had said that, okay, we have the sick building syndrome. Maybe it's not the mycotoxins, maybe it's the volatiles, and I had known about that. So now I'm going to get to talking about my work. Long introduction. So I hypothesized, hey, these Scandinavian guys, maybe they're right. The bad smell in my home might contribute to sick building syndrome. And as we have started doing this research, we have focused on a couple of 8-carbon volatiles, and we've used 2 approaches. We've exposed genetic model organisms to growing mold, and then we've used chemical standards of individual VOCs. And we came to doing this after years of studying the molds I'd isolated, and we found nothing new, no new species, no new volatile. But when we don't think anybody else has done this using the genetic models, and that's where we hit the pay dirt. So the one I have studied most, mushroom alcohol, simple 8-carbon alcohol, like many volatiles, it has enantiomers, and they have slightly different smells. To me, they both smell moldy. Theoretically, the S1 smells more like the moldy cheese, and the R1 more like the mushroom smell, but that's what it says in the literature. I can't really tell the difference. And we wasted a lot of time trying to find different activities for the 2 enantiomers, and basically they're both active. The R, which is the natural one, is more active, but they're both active. And it's found in human sweat. It happens to be the compound that attracts malaria mosquitoes. It's also known to deter a lot of compounds. And the first time I Googled the terms 1-octantriol plus insect, down came like 1,000 citations. There weren't that many in the mycological literature, but let me tell you, the entomologists believe what I say, not the mycologists don't always. And it's been, as I say, this compound's been used to attract malaria mosquitoes. There are close relatives that have been isolated as spore germination inhibitors and have been active in C. elegans tests. They have been studied for effects on humans, and actually there's a 8-carbon volatile that's used to inhibit essential tremors in humans. And so there's a big literature around these 8-carbon volatiles, but mycologists haven't spent much time studying it except as flavor. So my lab set out to say, okay, what were the fungi I found? What volatiles did they make? Can they be tested in genetic models? We've done pretty well on 1, 2, 3. We haven't done so well on finding out whether they cause sick building syndrome. And that turns out to be a big problem. What did I find in my home? Mostly trichoderma. This is because trichoderma is a soil fungus, and my house had been flooded with muddy soil. As I said, we didn't find any new compounds. We spent a lot of time isolating volatiles. It's not easy to do. We found that our molds made lots of different cocktails, but it was several years of not much new. And it's when we started using the genetic models that we hit the pay dirt. And here I give a lot of credit to R.T. Inamdar, who was a postdoc who led the project on the Drosophila fly. Sometimes in our studies, we expose larvae. Sometimes we expose adult flies. When you expose the larvae, you can watch them through an eclosion test and see how many pupate and how many turn into adults and how many have morphological abnormalities around it. With the adults, you can see how they behave, and you can also see whether they live or die, and you can count their dead bodies. We tried to do that with C. elegans, and they would just run away, and it was very unsatisfying because you couldn't find dead bodies. With Drosophila, you can see the dead bodies. And so here's some of the molds from Hurricane Katrina, and here is just some studies that are over 10 years old now, where we were able to kill a lot of Drosophila flies by just growing them next to trichoderma volatiles. And then we started studying these chemical standards, and the orange line is the mushroom alcohol, that compound that attracts malaria mosquitoes. And in a week, we could kill our flies by exposing them to fairly low quantities of this. Now, Artie had worked on paraquat, which is an herbicide in Parkinson's disease for her PhD dissertation. And she said, wow, these flies are behaving the way they do when exposed to paraquat. There's this fly model for Parkinson's disease. And so she started watching the way the flies moved. And sure enough, the 1-octantriol was acting the way paraquat does in causing these Parkinsonian symptoms in a fly model. And one of the great things about Drosophila, I've taught genetics for many years, but I'd never had flies in the lab, is it is a hugely well-studied genetic organism. So you can write to the Bloomington Stock Center and say, hey, send me a strain that has green fluorescent protein in front of my favorite gene. And they just send it to you, $27 or something like that. So we bought these flies that had GFP in front of the dopaminergic neurons and exposed the 1-octantriol, and you can see here's the downregulation of the genes. And then we got some flies that were transgenic, that had some special things in the fly genome that were overexpressing, and we were able to get rid of our effect. And I don't want to go into too much detail there, but we definitely were able to show that you can cause Parkinson's disease in a Drosophila fly, and have some very nice molecular data that shows that it was related to some of the genes involved in dopamine transport, and that one can think that possibly similar things are happening in mammals. I've written 3 NIH grants where I try to get funding to move this into mice or rats, and I get back reviews. She is not a neurobiologist. Okay, and so I've moved on to other things, but one good thing that's come out of it is I have a bioassay. And if people want to have some idea about the toxicology of fungi, you can use the fly model. And so we had our own hurricane in New Jersey, not that many years after Katrina. And it was a different kind of hurricane. It came late in the year. It was salty water, not Lake Pontchartrain. It was cold, blah, blah, blah. But I got invited to go out collecting molds after Hurricane Sandy. And yeah, I told you about the work with women in science. Even though there are a lot of women graduate students, look at who gets invited to go out collecting molds after Hurricane Katrina. And I was a senior scientist, and they sort of needed me. So I got to go out with the guys more than once. We brought a bunch of fungi back. We measured the percent mortality. Some fungal volatiles didn't kill the flies, others killed them a lot. We grew up the molds and tested the volatiles they're making. And one of the problems with this is it's very much a a snapshot. You measure the volatiles, and that's what you're getting at that instant with that fungus grown at that time. And I guarantee you, if you do 10 readings at different times, you'll get different things. But here's one of the fungi that was killing a lot of the fungus, and you can see that at 7 days, over half of the volatile it was making was 1-octantriol. And here's one of the non-toxic fungal volatiles that's not making any 1-octantriol at all. You can use this as a toxicology test. Anyway, the flies went well, and I'm going to be talking too long as it is, but we've been using these flies to study some medically important fungi, Aspergillus fumigatus, and done some very, very nice work with a recent graduate showing that Aspergillus fumigatus volatiles are toxic, and that we think some of them are acting as pathogenicity factors. I'm in a botany department, and I had students who said, well, we don't want to work with flies, so we're going to use another genetic model. And here's work done by Richard Hung, who was an undergraduate of mine at Tulane, and Samantha Lee, who's a Rutgers graduate all the way through. And the plant work is harder because plants need sunlight, and you have to use enclosed spaces. And we spent a lot of time developing different models. And you can see here this group one where we'd have a whole bunch of Petri plates on the bottom with the fungi growing and the plants above. And you always have to keep the fungus far apart from the target organism so that they're not touching. And that's the great thing about volatile stuff. No touching. It's all through the atmosphere. And we found that 1-octantriol can inhibit germination, and it can kill vegetative growth. And Richard Hung, I remember when he first sent me this picture, I thought that he'd made a mistake, because everything we had done to this point, the volatiles were doing bad stuff. And he sends me this picture, and I say, are you sure you have that right? Do you have the labels mixed up? And he said, no, I have it right. Turns out that fungi can make Arabidopsis grow better. And it's a series of biocontrol strains that have been used and known to do lots of things. Turns out that many of them make plants grow better when you're not touching the trichoderma to the volatile. And it took a long time. Samantha worked this out to figure out that we could put a little Petri plate with the trichoderma and have some plants on the other half. Otherwise, the trichoderma would just grow over and eat the plant. And it took about 2 years for this simple system to be developed. Once she did, we tested lots and lots of trichoderma strains. Actually, most of them made volatiles that made plants grow better. A couple of them, not so good. And I'm going to rush through this. And so she's got this whole wonderful collection of trichodermas from a guy named Kerry Samuels, and we grew them with the plants, and we measured the amount of chlorophyll, and most of them were improving it, and some of them didn't do much, and then grew all the strains and ran GC mass spec analysis, and lots and lots of work, and then looked to see which volatiles were being made by the different trichoderma strains. And here are the individual compounds that were tested, and then Samantha tested each one of these compounds individually to see whether they made Arabidopsis grow better or not, and whether there was more chlorophyll in the Arabidopsis, and found that some of them improved plant growth, some of them didn't do much. And then we only had enough money for one transcriptomic study, so we studied the volatile that had had the single best improvement under the conditions we used, and it was Decene. And we tested it at physiological compound, whatever, and we did a transcriptome study, and we found that there were only a few genes that were upregulated, and we'd really expected the auxin genes to be upregulated, and that's not what we found at all. We found that the major thing that Decene was doing was downregulating a lot of genes, and it was downregulating a lot of stress genes, which is interesting. So this plant, doesn't know that things are bad because its stress genes are downregulated, and it's growing better. And I had a visiting scientist from Iran who had been studying a strain over there and showing that volatiles were improving tolerance to salt stress. And so we hypothesized that what's happening And the reason these plants can grow better in the presence of trichoderma is that the volatiles are turning off their stress genes and they don't know they have salt around it. It may not be good for them in the long run, but they grow better. So trichoderma makes lots of VOCs. The volatiles change depending on the time, but if you study them one by one, you can break down and show something of their behavior, and at least with this one volatile, it downregulates stress genes. So one more model, yeast. Yeast has the opportunity of giving us a window on what's happening. Everything I've been telling you is phenomenological. I put in a volatile, I get an effect. Wouldn't it be nice to know what the volatile is doing? And we've made less progress here than some, but we're making progress. So, you know, yeast is even better than Drosophila. It's better than Arabidopsis. It's a fungus. And you can treat this particular fungus like a bacterium. So we've studied a whole bunch of different volatiles against yeast, and we have isolated some strains that behave differently, and we have tested the volatiles against a yeast knockout library. And you have to first find out what concentration of a given volatile will kill the yeast, and then you run it through the whole yeast knockout library and see which strains are still behaving the same way. And I didn't do this work, but we luckily in our building have a facility that does the yeast knockout library, and yeast is an even better model system maybe than Drosophila fly if you're looking for molecular events. And we went ahead and tested different compounds against the library and looked to see which genes were there. And this slide is perhaps the single easiest to describe, in which we are looking at the genes that were sensitive to 1-octanthreol, and that it's kind of backwards. It means that when these genes are absent, the yeast is able to grow at a concentration that normally is killing it. And so you have this kind of backwards things, that the resistance genes are somehow involved in enabling the yeast to grow, even though it would normally be killed. And up there in the upper left are the main ones of those. And they were screened to different types. And we found out that the single biggest category was involved in endosomal transport. And this work is not yet published because Shannon Morath, the PhD student who is working on it, dropped out during the pandemic. And so the work is on hold, and I'm very upset she hasn't come back. And we have all this beautiful data that hasn't been published. But the genes involved in the 1-octanthreol resistance have to do with some kind of sorting at the endosome. And we believe it has to do with getting the compounds into the cell. And when those genes are messed up, it doesn't get into the cell and it doesn't kill the fungus. So it's the first attempt that we know of to determine the mechanisms of what's happening with gas phase fungal volatiles, and that there are a lot of different genes that are turned on and off. And I didn't go into the details about tryptophan biosynthesis and ergosterol biosynthesis. I did mention the endosomal transport, and it's sitting there waiting to be written if Shannon Morath would come back from pandemic, and she hasn't. Okay, let me wrap this up. Volatile organic compounds are proving to be way more interesting and way more powerful than I expected. When I started this, I remember thinking, gosh, if I could find one effect. Every time I turn around, I'm drowning in effects. Every time I test a new volatile in any model, there are effects. And I started it because I was interested in sick building syndrome or building related illness. I haven't made much progress on that, but let me tell you, It's just opened up this whole world. Some of it's personal. I didn't pay much attention to smells until I started this. You know, I smell food or smell flowers or smell something bad. It was all hedonic. It was good or bad. I never gave any thought to why is that good or bad? Why? What is it that I'm sensing? And so now it's improved my world. It really has in many levels. It's made me think differently about all the years when I'd open an incubator and I'd smell something and I just ignored it. Oh, that's the fungi growing. Never paid any attention to why they're making that smell. Sometimes culture collections, these fungi that are in the deep freezer, they smell really weird. They're under a lot of stress. I've noticed that science is very much done in the liquid phase. The experiments we do, it's all fluid. It is, and we're just constantly throwing away the headspace. Almost everyone I know. I sometimes have trouble publishing papers because people don't seem to understand that's what's different about this paper is it's all in the gas phase, that we are not doing this the way most scientists do. So, hey, I'm acutely aware that I'm in the twilight of my career. And so my job is to try to inspire the younger generations. Look, in whatever you're studying, just pay a little attention to the things that smell around you, and you might be very surprised just how important those compounds are, and how frequently when people lose an effect when they're trying to do an extract, it may have to do with the fact that they've just ignored what's gone off in the gas phase. It really is an underexplored frontier in science. It's a very underexplored frontier in science. And it's not just for fungi, it's for all kinds of things. And I now kind of believe in aromatherapy. Unfortunately, aromatherapy books are a little too for me, but there's probably something there. Maybe lavender does do something to make us sleep better. You know, it hasn't been studied well, but there's almost something there. And then, you know, I'm more a microbiologist than a plant biologist. And when you teach microbiology, a lot of the textbooks start out about, oh, those people in the old days, they used to think that you got terribly sick because of bad air. And we know that you get terribly sick because of the malaria parasite or for the anthrax bacterium or for whatever. And aren't they stupid? But suppose some of the time, people were getting sick because there was something so nasty in the air. I mean, often old theories have some kernel of wisdom. And they say chemical ecologists have done some of the best work. And there is a division in biology between naturalists and molecular biologists that is often hard to breach. And the chemical ecologists and the entomologists have a lot to teach those of us who work on fungi. You know, we know that the stinkhorn smells bad. Maybe these compounds are doing some other things that we should know more about. And when people go looking for new medications, I mean, there are some medications that we inhale. I think there are probably some drugs to be found out there that are little molecules that could be, you know, drug companies should be looking for it. And we've all seen how dogs behave and the way they greet each other. And I think dogs can teach people a few lessons. I'm very, very high on the potential. Having said that, I want to remind you, right at the beginning, I mentioned that some of the volatiles that most people have heard of are volatile anesthetics. We've been using them for 150 years. We know what they do. They work. Do we know how they do it? And I've hung around some anesthesiologists, and there's some recent work on some volatile gases doing something to mitochondrial function and killing the mitochondria, but doesn't necessarily tell us what it's doing to our brains to make us not feel pain. And hey, just think of all that medical money and all those anesthesiologists. And here I am, this fungal researcher with a kind of big ego. Because remember, I told you we beat the penicillin people. I say, hey, maybe we fungal people can teach those who are working using the power of gases to learn something about what they are doing biologically. I found this slide. 3 forms of anesthesia, general, local, and what I'm doing, talking too long. So I'm going to wrap up. My favorite quote for being able to say thank you, he said it in French, Claude Bernard. Art is I, science is we. And it's interesting that, you know, we scientists have a reputation for being these isolated geniuses. We work together with people so much more than artists or writers do that it's a misplaced generalization. And none of us could get what we done if we don't work with others. And so I want to say thank you to some of my colleagues. And there's Shannon Morath standing in the middle next to the guy in the t-shirt, and she's the one who's sitting on all the beautiful yeast data and has disappeared during the pandemic. Jason Richardson, who is just in the process of moving to the University of Georgia, who helped with the Parkinson's work. Other members of the lab, and I had meant to put some arrows in here, but in the headscarf, Hadil Al-Maliki, who's done the beautiful work on Aspergillus fumigatus that I didn't tell you about, and Kayla Pennerman, who is front in the back, who has done a lot of our bioinformatics studies. And at the very end, I can't tell you how much it means to me to be here at Cornell. When I was an undergraduate, and you can see how old I am by this date, I won a NSF summer fellowship in the agronomy department here at Cornell in the summer of 1962. There am I, 40 pounds ago. And standing with a man, I can't remember his last name. His first name was Lacey. And I worked on a forage crop called bird's foot trefoil. And the experiments weren't much, but it was the first time I'd ever done research that was not in a formal lab. And so there was no answer. And I remember, wow, when I do this, it better be right because nobody knows what it's going to be. And that was very exciting to me. And here's picture of the agronomy department. It's a pretty male place. In fact, they had to build a special bathroom for me and the other woman who was out there in the field. And I reported to a guy named Robert Sini, who unfortunately I've written to him a bunch of times and he never wrote back. But at the end of the summer, he said to me, and I have it in quotes, but these are almost his words. He says, "You have a knack for this sort of thing. You ought to go to graduate school and become a cytogeneticist like Barbara McClintock." I'd never considered graduate school. I'm a first generation American. My parents didn't have much formal education. For me to go to college was a big deal. I'd never heard of Barbara McClintock. I applied to graduate school and Cornell was my first choice. Right after Christmas, I was rejected by Cornell. And I remember that. It was a bad moment in life. I had decided I was going to go to cytogenetics, following Barbara McClintock. However, it wasn't all bad, and I was accepted everywhere else. And the University of Chicago gave me a very nice fellowship. And I've never regretted going to the University of Chicago or the career that started. But, you know, I wouldn't have applied if it weren't for Cornell. And I have a special, I still love Cornell. And, you know, I can't say they did me a favor because I think I would have had a good career if I'd come here. But I had a wonderful career out of Chicago. And no regrets whatsoever. Hey, wake up and smell the fungi. I'm delighted to be here and thank you for listening.
Thank you very much for an amazing lecture. We thought that we will first give a chance to grad students to ask some questions. So are there any grad student questions over here?
Okay, I'm going to do a grad student thing that's going to embarrass them. I had meant to show a picture, but one of the students didn't send me an appropriate picture. But I have 3 former Rutgers students here, and I'd like them to stand up. Marianne, Andrea, and Jocelyn, all are students from Rutgers who are here at Cornell doing great things, and I'm delighted they're here. And so it's part of why I wanted to come is to see my former students thrive in the place that didn't let me in.
Well, if there are not grad student questions, maybe other questions here before we go to Geneva?
Yes, thanks for the talk. So my question is, so we always think there is a trade-off between plant immunity and growth. And you were showing the volatile compound will promote plant growth. Do you think those plants will be more susceptible to pathogens?
They might be. They might be. And I'm not a botanist, so I don't know much about plant immunity. Almost all the research I've seen has been about the saline resistance, and they definitely are growing better. And it's interesting, a lot of the work is coming out of the Middle East, like the student was from Iran, and there's some researchers in Egypt working on this. There hasn't been much molecular biology or phenomenology. It's just that, hey, it's happening. I don't know. And so you're going to need somebody who knows about plant immunity to answer that question. My sense is, though, these plants may be doing some other things right, because trichoderma is known to be such a wonderful organism to have in the soil. And I've focused on something that most people don't pay attention to, but trichoderma does a whole bunch of things. So if it's there in the soil, growing on the roots and touching the plant and improving other aspects at the same time, it's turning down some of the stress genes, they may be compensating. And if there's anything I have learned about biology, is that it's always more complicated than you think, always.
Thank you.
So, I have a question. Thinking about volatiles as a method of communication, how important do you think it is to elucidate like what the purpose of the communication is? Because, you know, you have different like eavesdropping between organisms,
Yes.
or versus like, you know, a co-evolved sort of interaction. How do we study that? And then also like, is it important to know whether or not these communicative signals are like supposed to be received by this organism, or if it's just like a
Well, not a good, it depends. I mean, there's so many signaling things that happen. So to make a generalization, yeah, I wouldn't even know how to begin. When you isolate a given interaction, which we do when we're reductionists, you're often leaving out a whole lot. And I am not ever going to be a good systems biologist. Because I'm trained in genetics, I tend to be very reductionist, but we need to do a better job of being able to study hundreds of things at the same time over time, and many organisms working together. And so when I'm giving advice to those younger and stronger, I say not only study the volatiles, but hey, learn a lot of bioinformatics while you're at it.
Laurie?
So I was really interested about the endosomal proteins. Do you think that the cells are importing, like it's explicitly importing the volatiles as a sensing mechanism? Or do you think that it's food or some other reason that it's active? Like, do you think it's active import?
Yeah, I don't know whether it's an active import or not. I am only at the very edge of this with this yeast work that there is definitely something going on at the membrane. Whether it's a passive or an active thing, do not know. It's certainly worth looking at. When I go back to the anesthetic gases, a lot of those are just diffusing through. And so one of the interesting things about small molecules like this is they seem to be able to do it without as much active transport. But again, there's just this big lacunae. We don't know. We simply don't know. And there's so many volatiles and so many things. And here, I've been very much focusing on 1-octanol, but I could focus on others and find other effects. And it's overwhelming. And as I said, I started this hoping to find, one or two effects, and instead I'm drowning in effects and spending the remainder of my life as this Johnny Appleseed character who's trying to go around and plant the seeds of future science in the minds of young graduate students and some of my colleagues to pay attention to these. So there are way more questions than answers, way more questions than answers.
Gary, you had one, and I'll open one on Zoom while you answer.
I enjoyed it. You talked about your work with the Trichoderma viride, and We see that very commonly as an ear mold on corn.
Oh,
And we've always put that in kind of a benign category as compared to the known mycotoxin producing molds
yeah.
on corn. Should we question that assumption now?
No, I don't
Especially
know.
going to animal feeds.
Yeah, I don't know one answer. I can't really answer on that one. Some of these trichodermas do make toxins, and certainly in feeds, we go back to where I started that I didn't talk about much. There's no question that some of the secondary metabolites fungi make are going to make animals sick. There's Verity, I don't know what kind of metabolites it makes, but a lot of trichodermas make some bad stuff. They're better in biocontrol than growing on your grain. Yeah.
Thank you.
Yeah, before we go to the chat.
Are these volatiles that stimulate plant growth, is that a concentration dependent phenomenon?
Yeah, there's a lot of concentration stuff here, and many of these things at low levels are doing good, and as they go up higher are toxic, and so
Yeah, okay.
and then you put in more than one. As I say, it is so complicated, but with my reductionist ways, we like taking one compound and trying it at a bunch of different concentrations. Often we're doing our experiments at concentrations that are not natural, because remember, you have to enclose stuff, and that's not how things are in the world. And so it's always a problem that we recognize that we have an artificial world when we do our experiments, but you've got to start somewhere. And that's where we're starting. But yes, they're very concentration dependent, very concentration dependent.
We have one in the chat that they ask, can you please expand on what is known about VOCs in the Penicillium species associated with cheeses? Should we be concerned if we store our camembert or roquefort in the refrigerator?
I do not know much about the volatiles of cheeses. I do know that they are a case of something that smells really terrible and tastes really good. And that's always been a mystery to me. It's kind of the reverse of coffee, which smells really good and tastes really terrible. And I wouldn't be particularly worried. I think that we have a lot of history of eating these cheeses with nothing but pleasure, but hey, they have found toxins from some of these Penicillia species mycotoxins, and we go on eating them. Everything in moderation, as they used to say. And but yeah,
And
it's a good question.
I'm
Stumped me.
Are there any more questions from Geneva or Zoom in general, chat, or just by speaking up? I don't think they are.
Okay, hey, thank you all for coming.
Yeah.
This has been a production of Cornell University, on the web at cornell.edu.